Hi everybody. Today we're going to make a foldable of a plant cell. So let's get started. So let's take any color that you want and let's just draw a cell membrane. Plant cells tend to be squarish, rectangularish. So we'll put that around there. And we'll call this the cell wall. And then I'm going to use another shade of green. And on the inside we'll make the cell membrane. Okay, that would be that one there. Let's go ahead and put a nucleus in here. We'll just go ahead and put it in the center of the cell, I guess. Nucleus. Let's go ahead and make this dark object called the nucleolus. Nucleolus, nucleoli, I guess is the plural. They stain quite nicely, and hopefully we'll be able to see that when we look at our cheek cells. Nuclear membrane would be this right here. And let's put a big old fat vacuole over here. Plant cells tend to have a large vacuole. Animal cells are kind of small. And then let's put a Golgi apparatus over here. There's just a bunch of tubes is what they are. And then rather than write in pink, I think I'm going to, I can find my pencil here, which I can't. I'm going to go ahead and write Golgi. You can call it Golgi body or a Golgi apparatus. I think your book calls it apparatus. I learned it body, so we'll just go with that. That's good too. Let's make a whole bunch of we'll call this the endoplasmic reticulum. Otherwise called the ER. And then on the endoplasmic reticulum often are found ribosomes. We'll just put a few of them here. There's actually an endoplasmic reticulum. They usually split it into two, called rough endoplasmic reticulum, and that's the kind that has ribosomes on it. And then the part that doesn't is called smooth. So we'll just go ahead and put some scattered ribosomes everywhere. So let's call this, these things here are ribosomes. Let's go ahead and put some mitochondria in the cell. And there's usually several. Mitochondria are the powerhouses, the energy sources, so there's usually quite a few. We're just going to draw 4 and we'll call that good. Mitochondria. Okay. Then let's go ahead and put, since this is a plant, we're going to put some chloroplasts in here. And they're typically bigger than the mitochondria, although they kind of have the same shape to them, but they're bigger and they're green. So chloroplasts everywhere. We'll just draw a couple so it doesn't get too crazy. That's a chloroplast. Okay, let's see what we're missing here. Nucleus, nucleolus, Golgi, vacuole. Oh, and then let's put in some, they're called lysosomes. We'll just make them this color. They're usually just circular things that are kind of darkish looking. And we'll just, there's a bunch of them in there, so we'll just put that around. Okay, so let's call, oh, I guess this one, lysosome. There's debate whether lysosomes are found in plant cells, and we've drawn it in a plant cell, so I'm going to go ahead and put it in here because the latest research seems to think that there are lysosomes in plant cells. But definitely, the way I learned it is that they were only in animal cells, but we're going to go ahead and put them in this plant cell as well. Okay, so I think we have everything that we need on this foldable. Okay, so now we're going to make some flaps, and the first flap we're going to make is the mitochondria. Okay, there's a flap for that. Mitochondria. Let's make a flap for the ribosomes. Alrighty. And then we'll make one for the chloroplasts. And for that one, we're going to fold the chloroplast name in half and cut underneath it. And from that point, we're going to cut up. Make it a little bit longer here. Okay, and we'll go up like that, and that'll be the flap for chloroplasts. And then we have the cell wall. And we have the cell membrane. You can make your cuts anywhere, by the way. You don't have to do what I do, but let's see here. So we need a flap for the vacuole, so we're going to put that one in the middle. Let's go ahead and fold the vacuole name in half. And then we'll go like that. We'll make a flap up that way. Okay, that's our flap for the vacuole. We'll make one for the lysosome. Flap going that way. And the Golgi body gets a flap as well. Fold that name in half. Cut that way. And then we'll come back up like this. And then like this. And fold it up like that to get that flap for the Golgi body. The ER, endoplasmic reticulum. I'll make that a center flap as well. And let's go like that. And then we'll go straight up for that one. Straight over for that one. Flap up. Okay, let's see. Who in the nucleus needs one? Well, let's do the nucleolus first. A lot of flaps. Okay, there's one for the nucleolus. And then we got one for the nucleus. There. So, there's something that we're missing, though. Did we get lysosome? We did. Oh, I know what it is. The cytoplasm. Cytoplasm is everywhere. It's a jelly-like material that all of these organelles sit in. The cell doesn't have any empty space in it. And so all this white, everything that we've left white, is not empty space in between the organelles. Scientists for the longest time believed that cytoplasm was just jelly-like material that held these organelles in place. And while that's true, in the last several years, they've discovered that in this cytoplasm here, there are a lot of chemical reactions taking place inside of the cell. So everything that you see, all these organelles, sit inside of something called cytoplasm. And we got to find a place to put cytoplasm. Cytoplasm. There we go. Okay, so we'll just leave it like that. All right, so now you're going to take your foldable, and on the back side you're going to put a bunch of glue, but don't put the glue on the flaps, just in between. And then you're going to put it on your piece of construction paper, like so. And then we are going to start writing underneath the flaps. So let's just start off with the cell membrane. Okay, the pencils, put them over here. The cell membrane controls materials that leave and enter the cell. Controls materials that leave and enter the cell. Okay. Then we have over here on the right the cell wall. Two purposes. One is to protect the cell, and the second one is to give the cell strength. So cell walls also allow plants to be somewhat flexible. If plants are growing outside and it's very windy and hail is coming down, a lot of trees will bend without snapping, but yet they're very sturdy in that they can grow straight, towards the sun, and so that would be the cell wall. Gives them strength, and it's because of where they live that they have that in their cells, and animals, of course, do not. Chloroplast, the job of the chloroplast is to capture, it captures sunlight, and changes its energy to a usable form. And that's because plants make their own food. So they use the energy in the form of light from the sun, and then the chloroplast changes that energy to a form that they can use so that they can make their own food. The job of the ribosome is to produce proteins. Now you know where those proteins come from that make DNA. The job of the mitochondria is to convert energy in food to energy for cell use. So plants make their own food, and so they make their food for the same reason you and I do, so that they have energy. And so it's the mitochondria that takes the energy from the food and converts it to a means that the plant can use it for its daily activities. So the job of the ER, the endoplasmic reticulum, it's threefold, and I'm going to write on the inside of the flap because I don't think I'll have enough room. Number one is that the ER produces substances that the cell needs. The second thing it does, there are some ribosomes attached to the ER, so the ER helps them produce protein. And the third thing that the ER does, oh, you know what? That's the second and the third thing. The second thing is that it produces, okay, so I goofed up. We'll just switch them around. This was actually number 2, but we'll make this number 3. They actually produce protein. Okay, so let me go over this again. Number 1 is produces substances. We got that. Second is that there are some ribosomes that are attached. Very good. And the third thing I was putting in with number 2. That's what goofed me up here. The third thing is that right here, it helps them produce protein. So you can make that number 3 down here if you want. Okay, very good. That's the endoplasmic reticulum. Then the Golgi apparatus over here, okay, its job is to package and distribute materials from the ER, endoplasmic reticulum. And that's why the Golgi body is typically found pretty close to the ER. I probably should have drawn it closer here, but so it takes all of these materials that are produced in the ER, and then it distributes them throughout the cell wherever it's needed. And the way it does this is through all these tubes here. So it's kind of like a bloodstream with arteries and veins. All these tubes are the transportation system, if you will. Okay, the lysosome. The lysosome is considered to be the garbage collector of the cell. The lysosome will break down large food particles into smaller ones. Okay. It's also, like I said, considered to be the garbage dump. It also gets rid of some things that wastes that the cell doesn't need anymore. The vacuole is the storage area. It stores water, food, waste, other things, etc. Just put etc. down. That'll be good. Okay, so what do we have here? We cell membrane, I think, oh, nucleolus. Nucleolus. Let's go to the nucleolus. Nucleolus. Ribosomes are made here. So ribosomes are made here. Once we have the ribosomes, they can make protein. Once we have protein, then we can get some DNA. And the nucleus directs the cell's activities. It's the brain of the cell. Okay, so nuclear membrane, of course, is just like the cell membrane. It controls what enters and leaves the nucleus.